that probably has more longevity power than any dietary component.
I am a super young person studying a super old person.
She was older than me during World War II.
Maybe we have to change that paradigm.
That is a really astute point that I didn't really think deeply about.
It's not about luck, after all. Maybe it's more about habits.
Maybe we don't have to die at like a normal age, 120. Maybe we can engineer to live to 500, 1,000, or make death optional. Dang, LDL cholesterol finally got her. All right, everybody, we're back with another interview, and this one is something special. But just to give it a little bit of framing, human longevity seems to feature more and more in headlines these days. I'm sure you've observed that too, and it makes sense. It's an area of immense public interest. And sometimes the headlines are a little gimmicky, and then other times they refer to studies that knock your socks off. So today I'm joined by Dr.to-be Eloy Santos Pujol. I hope I pronounced that right. He's a PhD student in Catalonia, Spain, whose work focuses on the biology of aging. And he was the lead author, the first author on a widely circulated study published in Cell Reports Medicine. I have it printed out right here on a supercentenarian, a woman who passed last year at the age of 117 and 168 days. I was trying to wrap my 29-year-old head around that, and I realized she was older than me during World War II, which is kind of mind-blowing. And I'm thrilled to have Eloy with us here today to discuss his paper. And I also just want to know, a lot of the questions that I'm going to bring up come from our Stay Curious community at staycuriousmetabolism.com, the newsletter. So thank you to everybody for putting in your questions. That pass a lot of them on to Eloy today, and we can get his 2 cents. Eloy, thanks for joining me.
Thank you for inviting me.
So before we get into the nitty gritty, I just want to ask, so you're a PhD student. Having been a PhD student myself and having known many, I have to say this is a really immense project in scope and impact for, a PhD project. How did this fall to you in the first place?
Well, we were already researching into longevity and human aging, and suddenly it came up to us that now the world's oldest person in the world was then in Catalonia, right next to us. So it was like, okay, now it's a very good opportunity to continue our research in now the world's oldest living person. And yeah, that's how it came to us. It was a good opportunity for us to get hands-on on the most extreme longevity, and also to have some clues on how the oldest people, these supercentenarians, these rare people, can live and get to that exciting age.
It's a very exciting project. What did it feel like to take it on? Was there like a moment when they're like, hey, this is now your project?
Yeah, well, it was like a responsibility for me, because researching into the world's oldest person is something that not all people can do. It was like, for me, like too much, and yeah, I have enjoyed it a lot.
Well, your paper is absolutely phenomenal. Everybody should try to go read it. It's really well written. Before we get into Maria, I just want to ask a little bit more about your background. First, how old are you?
I'm 26. I am a super young person studying a super old person.
I think that's fascinating how, our generation, so I'm 29, is interested in metabolic health and longevity. And I think it captures something about changing society. I think people say death is part of life, but I think we're kind of getting to a place in science, medicine, and society where we're kind of just thinking in the back of our heads, maybe not articulating it, but maybe we don't have to die at like a normal age, 120. Maybe we can engineer to live to 500, 1,000, or make death optional. That's my 2 cents. I do want to ask, what got you interested in the biology of aging?
It's like an investment. I think that when we are young, we start thinking about economic investments, maybe getting a house or getting a car to go to work. I think that it is the most important investment someone can do in their life, to start earlier, maybe in their 20s, 30s, to take care of their health. And if they can, and they have the luck that they had to do some active research into it and help the world get to healthier lives and longer lives if possible.
Yeah. Well, I love your enthusiasm. And just to reiterate something that you were telling me before we started, the way you explained your interest in longevity, you were saying something to the effect of, and you can reiterate if I'm misquoting you, but we study all these things in silos: brain disease, Alzheimer's, cardiovascular disease, and they're all aging phenomenon. So you're like, why not just go to the root phenomenon? What can we uncover that is common to all these diseases? We're going to get into it later, but I just wanted to tee that up for the audience because I thought like that's how I think about metabolic health. You could call it longevity science or whatever, but it's the roots of the tree of disease, which we so often overlook, a siloed medical system. Actually, on that, before we progress, I have experience training in the medical system in the United States. How is it in Spain? Is it similarly siloed, or do you feel like there's any more integration?
Well, yeah, and it has to do with what you were talking about. All diseases have a single treatment. They are treated like everything has a drug, a different drug, and a different research group researching into that disease. Okay, maybe we have to change that paradigm and maybe go to a future where we can research aging and then not only prevent those age-related diseases, but also treat them. We'll see where it's coming.
I agree. And there's one particular example that jumped up in the paper around cholesterol that I know we're going to get into. But before we get to that, Maria. So Maria was the supercentenarian's name. What do you know about her? Did you ever have any interactions with her personally?
Yeah, sure. I visited her several times to get some samples. She was super cute. This cute old woman that everybody would like to meet somewhere in life. And yeah, it was a pleasure to meet her. It was special how well she could reason what she was saying, or how she could remember past events, how lucid her mind was, how sharp it was, and also how kind and generous she was to give us some samples to study her.
Yeah, that's really amazing. It didn't come through in the like I'm not sure you can calculate that in the paper, but it sounds like at the end of her life, she was still completely there, which at 117 is really incredible. I also heard that she requested to be studied. Somebody quoted her as like, "Please study me." Is that an accurate representation? Was it her request?
Well, I think that the first request came from us. And after that, she was willing to collaborate fully with us and even told us that she wanted to be studied by us. I think that she knew that we could get a lot from her. But she was very humble, and all the time she was saying, well, the only merit that I have is that I have lived like too long. Just that I haven't done anything special. She was very humble, but
Yeah.
yeah.
Well, maybe we can get into more of her background later, but before we get into the biology, I find with all these stories of supercentenarians, they're all pretty diverse in their diet and lifestyle, but they tend to have maybe similar mental or cognitive traits. Were there like personality traits? You alluded to a few, but that really stood out for you in interacting with Maria?
Was very positive and optimistic, and she was always surrounded by the ones that she loved: friends, family, and even dogs. She loved dogs, and I think that this social part of longevity, which is very poorly studied, is something that also contributed to her longevity.
Yeah, I would tend to agree. It's one of those things. Maybe there'll be follow-up research. That's not on her; she's passed. But like, how concretely does something like social interaction, something that seems so abstract, or like mindset, impact biology? And just to kind of now go on my own little soapbox and tangent, like there are studies coming out. I can send you this one I just read. I think it was in Nature, about like how a psychological stress can like basically make your liver make sugar and change your metabolism. Like there are direct lines between the brain and the liver and other organs that like it is hard biology. It's just also very hard to study because, the mind is an emergent phenomenon of the brain. How do you operationalize all this? So that's a topic for another time, but you hit the nail on the head. I tend to agree. There is something about mindset, attitude, social connection that probably has more longevity power than any dietary component. But with that, that's still an area of interest, so we'll dive into it. So I want to get into the data. One striking result to me was her telomere length. Her telomeres were tiny. Tell us about that.
Well, in her case, I think that the telomere shortening, this extreme telomere shortening, was like a proxy of how old she actually was. It was like something that really revealed how old her cells were. Maybe it serves as not only predict mortality or whatever are associated with susceptibility to diseases. Well, in her case, I think that it was just a very clear and accurate prediction of her close death. I think that in this case, telomeres were not reflecting any health-associated trait, but just this chronological trait in her cells.
Yeah. Let me double click on that and clarify, because I think I wrote a letter on it and then did a video coverage, and that was one of the most confusing things for people. So I'll show a graph on screen now, which is where Maria's telomere length, she's the orange dot, tracks with the general population. And what you can see is it's like right on line with the general population, exactly what you'd expect. So to your point, it's like telomeres, which, just to define them, they're these little caps of repetitive DNA on the end of your chromosomes to kind of protect genetic material. As cells replicate, you kind of lose the caps. So they protect the important genetic material. And as they're kind of like the aglet at the end of a shoelace. And as they shorten, the idea is like chromosomes tend to fray. You get, it's thought to be a proxy for biological aging. And I really want to emphasize that. And my expectation would be that if Maria is aging slower than the average person, such that she can live out to 117 years, that her telomeres would be shortening at a lesser rate, rather than having the telomeres just be like a clock of her actual chronological age. So by analogy, it's kind of like rings on a tree. The number of rings tells you how old the tree is. And in this case, that's kind of what happened for Maria. So my expectation wouldn't have been that she falls on a population distribution, that her telomeres are shortening at an average rate, just strung out to 117, but that's what happened. Now, the reason I'm really delving into this is because, I don't know if you agree, but it's suggested that telomeres aren't actually that important as a marker of longevity and health span. Whereas before, I think we thought they were. What do you think?
Well, this is a study made in only one person. So to what extent can we conclude that? I don't know. Maybe just in this person, telomeres were not reflecting her younger biological age, but just the chronological one. And maybe that would be different in all the supercentenarians. But in her case, maybe her longevity, or at least this healthy-like phenotype, might not be coming from telomeres at all. Telomeres are only one of the hallmarks of aging. It's like there are different sources of youthful traits. Maybe this was not the case for telomeres.
Yeah, to your point, a caveat, it's only an N equals 1. And there's other possibilities, like maybe at the very end of life, her telomeres shorten quite rapidly. But I did find it an interesting thing that I wanted to delve into a little bit. So you mentioned other hallmarks of aging. What stood out in Maria as maybe a very important hallmark of aging that contributed to her longevity?
In my opinion, it was microbiome and epigenetic age. Epigenetics are the chemical marks that are deposited onto the DNA that are controlling gene expression. In her case, these marks resemble those of younger people, even up to 15 or 20 years younger. It was like too much. And at the beginning, we were like, okay, maybe we committed a mistake at analyzing these results. But no, it was actually like this. She had an epigenetic age much younger of what hereditary art was revealing. And it was like, okay, well, it is possibly one of the pathways that were favoring her extreme old age and healthy quality of life. Also for the microbiome, we could see that there was a strong population of bifidobacterium, which is quite beneficial and has already been seen to be linked with longevity, lower levels of inflammation, and also this prevents the growth of damaging bacteria in the gut. So maybe the contribution of these two hallmarks, together with a good metabolism and other features that we have found in her, might be contributing in a greater extent maybe to this extreme phenotype.
Yeah, I kind of threw you a hardball there, because with something like this, such a treasure trove of data, other things that stood out to me were her mitochondrial health. There was a line in the paper, you probably wrote it, about her mitochondrial health was not only preserved, but robust. And then her engaged lipid metabolism and other markers too, like she had very high lactate and creatine levels. There's so much to delve into. But just to take a step back and think about what might have been some underlying factors contributing to all of this. So epigenetic age, mitochondrial health, good gut health. We'll get into diet in a minute. But do you think that there were other features of her life that contributed to it beyond just luck and genetics?
It's not about luck. After all, maybe it's more about habits, because that's what is mostly influencing both epigenetic age and microbiome. Especially for epigenetic age, it might be contributing the way you sleep, whether you are doing exercise, whether you are socializing, actually, or also for the microbiome. Everybody knows it's tightly connected to the way we are eating. In her case, she was keeping a Mediterranean diet, rich in fiber, yogurt, and supplemented with protein. Maybe it was all this combination of habits together with those special variants that we found in her genome, which would have driven her to this special longevity and good quality of life throughout her life.
I want to delve into that and forgive me in advance for some yes or no questions or a lot of things that my audience wanted me to follow up on. But first on her diet. So I looked at her meal plan. Thank you for providing that. It's like rich in eggs, egg protein, olive oil, probiotic yogurts. My first question is, do you know why she followed this particular pattern?
Well, it's the same meal plan that it's given to all people living in that residence for the elderly. Maybe diet was something very common in that place for everybody. It's true that it was special for her, the fact of eating so many yogurts. She loved it, and of course the people in the residence couldn't say no to her at her age. So they gave her all the yogurts she asked, and after all, maybe it could have contributed actually to this enormous population of bifidobacterium. We don't know. We'll have to validate it with larger cohorts and see whether this healthy population might be actually contributing that much to longevity.
How about anything about her activity? So we mentioned her social connection. Was she an active person? Did she like exercise?
When she was young, she could do exercise. She went walking with friends, also socializing while doing exercise. This is something very, very interesting to think about. Also, she played tennis, she played with the dogs, she kept a garden. Well, she was active. And she kept being active until the end, until she could. There was a point where she couldn't move too much and she had to stop doing exercise. But at the end, she still had very good sleeping patterns and diet that might also help make up for that absence of exercise during her last years of life.
That's really impressive. How about, was she on any supplements or medications?
Well, just supplementation in egg with a protein to make up for sarcopenia, which is very common in old people. This loss of muscle during time. And that's all. When she had some fever or some infection by bacteria, well, I suppose she got all the drugs necessary for that, but not chronic drugs, these kind of drugs that you are consuming every day.
Wow. And did she have any like medical comorbidities, any history of heart disease or anything?
Well, diverticulum disease, some vertigo, the hair loss, disease that appear with age. I would actually be surprised if she wouldn't have all these problems, at least one of them, at the end of her life.
So it doesn't sound like any like proper diseases, just general declines in organ systems, which, you have a little bit of hearing loss. But otherwise, she sounded pretty dang healthy. Any alcohol intake or smoking history?
Very small quantities of alcohol in very special occasions, and no tobacco, no smoking at all.
Well, I now wanted to get on to something I found particularly interesting. So in the supplement, you mentioned her LDL cholesterol and her LDL particle count. Both of these were actually above the recommended range. Now, they weren't astronomical. I guess I'll ask a provocative question first. Why were these put in the supplement and not the main text?
Well, the LDL cholesterol is in the main figures. It's in the figure 2 where there's a cycle summarizing all the parameters contributing.
So it was like marked as red as quote unquote high.
Exactly. That's a good summary of her cardiovascular disease measured by those different parameters. And the LDL cholesterol, you're right, it was a little bit increased in comparison to the general population. It wasn't that much. I think that the threshold was at 100 and she had 120 something. It wasn't that high. But all the other parameters also contributing to that cycle, summarizing the cardiovascular disease, were okay, specifically esterified cholesterol, free cholesterol, HDL particles, which is a good good cholesterol, which was quite increased in comparison to the rest of the population. I think that we have to take into account all those parameters in order to have a full picture of what her cardiovascular disease risk was. Well, this is true. LDL cholesterol might be important for some diseases and health conditions, but taking all into account all the parameters related with that, I don't think it was that high and that it was contributing that much to the overall disease risk or all-cause mortality risk.
100% agree. And there's a few things that you said, particularly the whole picture, that I just want to emphasize. And I'll disclose the reason I asked this question of you is because in my experience, both as a scientist and a medical trainee, that marker, the LDL cholesterol, takes up so much airtime. It gets like keyhole focused on, and generally in a very black and white manner. So when you get like your lab report back, there's a threshold. In this case, you mentioned it's 100. And if you're above that, you're in the red. And then the biomarker gets treated, but not in the full context. So I totally agree with the way you framed it up. It's like, look, she has great overall metabolic health. You use the term engaged with the metabolism. There's markers of that. Her high HDL cholesterol and high HDL particle count, very low triglycerides and very low triglyceride-rich particles, VLDL. So the fact that her LDL was elevated in this context, I don't think has much bearing on her cardiovascular health or her longevity. So I very much agree with the way you framed it and the fact that those markers were de-emphasized. The reason I'm bringing it up is not to give you a hard time, but to juxtapose it to at least my experience with how conventional medicine is practiced, where it's like you have the marker that you know how to treat. In this case, LDL with, say, statin drugs. If it's out of range, you just hit it with the drug, and it's not taken into the fuller context of other metabolic markers in the patient. That's why I wanted to bring this up. I don't disagree with it being, that number being de-emphasized to an extent, but, I thought it was relevant. Anything to add on that before I ask you your next set of questions?
Actually, I don't think it was the main reason she died.
I don't. So actually, I think I dropped a video yesterday. I think the top comment, which was said in jest, I'll pull up the screenshot here. Somebody says, 117, that dang LDL cholesterol finally got her. As just like a joke to be like, there are goats. Anyway, I found that funny. That had me laughing out loud. So moving on though, her genetics. So you mentioned a ton of gene variants. What stood out to you in her genetics?
It stood out to me some genetic variants that were found in homozygosis, which are present in both alleles, the one coming from the father and the one coming from the mother, which are the ones that may be contributing strongly to her phenotype. Some variants related with cognitive function, also heart function and immune function, and especially those variants in the mitochondria, which are only inherited from the mother. So they might be contributing somehow to that robust mitochondrial function that you were mentioning earlier.
Were they, so just for people's background, your mitochondria, quote unquote, the powerhouse of the cell, they had their own genetic material, their own DNA. I forget how many genes there are in that, but they also have a contribution of nuclear DNA. So the main kind of host genome. Were her particular mitochondrial variants predominantly from the mitochondrial DNA or the nuclear DNA?
There were 3 from the mitochondrial DNA and a couple or 3, I don't remember now, that were codified in the nuclear DNA. So it was a combination of both.
Okay, cool. Yeah, that's super interesting. I also actually, now tangenting a little bit more, one area of active research, I was interviewing Professor Michael Snyder, and he's consulting for a company called Matrix. I have no affiliation, but is mitochondrial transplantation. So ex vivo generating mitochondria en masse and then transfusing it into people. So yes, this is n equals 1, but my mind is now worrying because when we're talking about longevity science, you can't edit a human's nuclear DNA, but you can transfuse them with mitochondria with their own DNA, youthful DNA, and mitochondria actually circulate around in the body in small extracellular vesicles and free mitochondria. That's a whole other topic, but provides a lot of hope for actual medical interventions that could expand longevity and health spans, in my opinion. So exciting stuff. And a couple markers people were asking about. Did she have an Lp(a) per chance in her lipid profile anywhere?
The lipoprotein profile was the GlyK, you mean, huh?
There's something called the lipoprotein(a). I didn't note that marker. I didn't know if it was anywhere. My assumption was it was not, but I got that question and I looked and I couldn't find that marker. I don't know if it's something that we were able to measure, are there any biosamples? It's another marker of cardiovascular risk. They're kind of like LDL particles, but they have this little tail that makes them more clotty. I can send more information if it's not something you're familiar with, but it was just an area of interest for those interested in her cardio metabolic health.
Sure, I agree. It might be super interesting, but to be honest, I'm not familiar with this. I'm not so focused on metabolism like you. I'm not that expert. I'm more expert in genetics, epigenetics, and yeah, I'm not that familiar with that protein, but I will have a look at it. Sure.
And did she get an insulin level at any point?
No, we couldn't measure that, but we know that she didn't have any diabetes going on, especially type 2 diabetes. We asked the clinician, she didn't have any disease related with diabetes, hypertension, and all the stuff that you are often checked in the clinic. In her case, apparently everything was okay, and there were some metabolic variants that could reflect the absence of high levels of glucose in the blood. For example, the GLIC-A and GLIC-B, apart from being markers of inflammation, they are markers of high sugar in the bloodstream. In her case, those proteins were very low, and we can see that she wouldn't have a lot of quantity of sugar in her bloodstream, I'm sure.
I agree. I think based on all the mountain of data that were provided, one can intuit she doesn't have high insulin, hyperinsulinemia, or insulin resistance. I think it's a fair conclusion. Kind of getting towards the end, is there anything people you feel are misunderstanding about Maria's case or this paper that you want to clarify?
Well, actually, maybe people is thinking that yogurts may be the measure contributing in longevity. I could see ads reporting the brand of the yogurt that she was eating. This measured presence of yogurt in her life. Well, maybe it's not the case. Maybe it was just like a help that she had to have a good microbiome, but the microbiome was not the only thing that had helpful traits in her. It was also the epigenetics closely related to our habits, and also that those genetic variants that we could see related with cognitive function, heart disease, and metabolism, also with immune system. We saw also a variant called timeless, which has already been described to be contributing to longevity in Drosophila. I mean, there were a plethora of things going on in this person to help her live to the 117 years of age. There's something that nobody is talking about. It's the death cause. We don't know. When someone that old is dying, nobody asks whether she died, well, for some reason in particular. Everybody takes for granted that she died from being old. But what does it mean? What does dying from being old actually mean? I think that this is something that scientists will have to explore in the future. If we had some clues about what drove her to succumbing to death, maybe we would be having some clues to do some interesting research in the future. We will see. I think that this is something very, very interesting to talk about, the real death causes of people dying super old.
That is a really astute point that I have to admit I didn't really think deeply about. And I think now thinking about it, we have just internalized this idea that you can die of old age. But it's a vacuous, hand-wavy thing.
Yeah, what does it mean?
So yeah, what took her? That's
exactly
a really great point. That's fascinating. I don't know. Well, you're the guy who's going to figure out the answer. So what's next for this line of research? So you're early on in your career. You're 26 doing your PhD. Do you have a new project that's chasing this up, or areas of research you're interested in?
Now we have to look at the daughters and her grandsons. I think it would be interesting to look at her family, how the genetics variants were inherited in her genealogic tree, and see whether it also had a role in their epigenetic age, telomere attrition, and all the stuff. I think that now it's time for us to look at her family.
How many kids does she have? And grandkids, and great-grandkids, and maybe even great-great-grandkids. Her family tree must be crazy.
She had 2 daughters and a son. The daughters are still alive, and we'll study them for sure. And regarding grandsons and grand-grandsons, I don't know how many, but she has a lot.
That's really incredible. All right. Is there anything I should have asked you, Eloy, something about Maria, the paper, anything that you want to share.
Well, I think we talked about everything more or less. So, and it was very interesting.
I really appreciate your time. Is there anywhere you want to direct people to other than the paper, which I'll post below? Your socials or research pages.
Yeah, they can follow me on Twitter, Eloy Santos Pujol, also Instagram, Ivael. And well, stay tuned because our research won't stop here and we'll continue looking at the family and also doing different studies to help understand longevity and human aging.
Amazing. All right. This is definitely a guy to follow. Going to be a leader in the longevity field for the next 100 years until somebody studies you and you pass at 126. Anyway, thanks for joining me. And yeah, I'm sure everybody appreciated this conversation.
Thank you, Nick.